interim clinical data from the Phase 1 study of NTLA2001. This conference is being recorded at the company's request and will be available on the company's website.
Today's call. Dr. John Leonard, our Chief Executive Officer, will begin with brief introductory remarks. Dr. Julian Gilmore, Professor of Medicine at the National Amyloidosis Center and physician at the Royal Free Hospital in the UK, will review the interim results. Dr. Gilmore is a respected key opinion leader on the treatment of amyloidosis and the national coordinating investigator on our clinical trial in the UK. Dr. David Ledwalla, our Chief Medical Officer, will then speak to the next steps in the clinical development of NTLA2001. And Dr. Laura Sepp-Lorenzino, Chief Scientific Officer at Intellia, will outline the implications of these results, both for our proprietary LNP-based platform for systemic gene editing and for our pipeline of in vivo therapeutics in research and development. John then will return for closing remarks before we open up. Intellia undertakes no duty to update this information unless required by law. With that, let me turn the call over to our CEO. John?
Thank you, Lena, and welcome everyone. Today, we're incredibly proud to share positive interim data from Part 1, the dose escalation portion of our Phase 1 study evaluating NTLA2001 as a potential one-time treatment for transthyretin amyloidosis, also known as ATTR amyloidosis. NTLA2001 is not only our lead candidate, but also the first systemically delivered CRISPR-based therapy to enter clinical development. That is, the first CRISPR-based drug candidate to be intravenously administered and edit the human genome in the target cells inside the body, the basis of our in vivo approach. As such, the study is intended both to evaluate NTLA2001 as a potentially curative treatment option for ATTR amyloidosis and to validate our in vivo editing and delivery platform. Over the past several months leading up to this announcement, we viewed this readout through three critical lenses: what it means for patients, what it means for Intellia, and what it means for the field of medicine. For patients with ATTR amyloidosis, NTLA2001 is the only treatment with curative potential that may both halt and reverse disease progression with a single dose. Despite the availability of chronically administered therapies, there remains unmet need in patients with ATTR amyloidosis. We believe a single dose therapy that produces greater TTR reduction will offer significant benefit to patients and value to our healthcare system over the current standard of care. For Intellia, this is a profoundly important milestone. We've worked hard to advance our platform to the point of clinical validation. We recognize that these results are significant to our broader in vivo pipeline development efforts by helping to establish proof of concept for our modular approach that we incorporate into future programs. This announcement is our first clinical data readout, and for that alone, we're grateful to our team and partners at Regeneron for their shared and tireless commitment to bringing our technology to patients. But more than that, we're grateful to the patients who are participating in the trial. And for the field, as the first ever clinical data supporting the safety and efficacy of systemic in vivo CRISPR genome editing, we believe this opens the door to a new era in medicine. We recognize these are interim Phase 1 results, but we believe CRISPR-Cas9 and genome editing more broadly is an immensely powerful tool with vast potential impact. And these results begin to demonstrate that. Moreover, our technical achievement of systemic administration marks the beginning of bringing CRISPR as a beneficial therapeutic to broad patient populations. This work is central to our mission to advance curative genome editing treatments for patients with severe diseases and is why we believe these data represent a momentous milestone for medicine. At Intellia, we've sought to harness the power of CRISPR as a therapeutic modality by developing in vivo and ex vivo approaches. For genetic diseases, we've utilized an in vivo approach where CRISPR is the medicine. We leverage our proprietary non-viral lipid nanoparticle-based CRISPR delivery system to selectively inactivate disease-causing genes or precisely insert genes to produce normal proteins. And although not the focus of today's call, our ex vivo approach, where CRISPR creates the medicine, is designed to edit genes from patient-derived cells for treatment of immuno-oncology and autoimmune diseases. Implementing this full-spectrum strategy, we focused on building the broadest, deepest toolbox in the genome editing space, and we've developed a modular platform that we can leverage from one program to the next. These efforts have produced a rapidly expanding pipeline aimed at extending the benefits of our Nobel Prize-winning CRISPR-Cas9 technology to the broadest possible population of patients. This is, in part, what makes the platform lens of today's readout so meaningful, and enables robust expansion of our pipeline over the next several years. With all this in mind, we're pleased to share our interim clinical data from our presentation at the Peripheral Nerve Society with you today, and we note the publication of this work in the New England Journal of Medicine on Saturday. Based on interim data in 6 patients from Part 1 of our first-in-human study evaluating NTLA2001, treatment with NTLA2001 led to a dose-dependent response in serum TTR reduction. In our second dose level, we achieved an average TTR reduction of 87% at day 28, including one patient with a 96% TTR reduction. These deep TTR reductions support our view that NTLA2001 has clear potential to deliver life-changing benefits for patients with ATTR amyloidosis. Importantly, we observed encouraging safety and tolerability for our proprietary lipid nanoparticle, or LNP, delivery technology in RNA CRISPR cargo in these patients with no serious adverse events in the first 6 patients by day 28. Altogether, the data suggests that we can achieve targeted in vivo editing for desired pharmacological effect. And although there's still much left to learn from the study regarding this program and our platform, we believe these results substantially de-risk our in vivo efforts, validating our non-viral approach to systemic delivery and unlocking treatment of diseases that originate in the liver. David and Laura will touch more on the implications of these results shortly. For now, it is my pleasure to welcome Dr. Julian Gilmore, who will take us through the data in detail and offer his perspective as the National Coordinating Investigator on our Phase 1 trial in the UK and a leading physician specializing in treatment of ATTR amyloidosis. Dr. Gilmore, over to you.
Thank you very much. So for listeners who may not be familiar with transthyretin amyloidosis, also known as ATTR amyloidosis, it's a rare, progressive, and fatal disease caused by the accumulation of amyloid deposits which are composed of misfolded transthyretin or TTR protein. The disease may be hereditary, caused by a mutation in the TTR gene, of which there are now more than 120 known. Hereditary ATTR amyloidosis is estimated to affect roughly 50,000 individuals worldwide and may predominantly affect the nerves, causing polyneuropathy, or the heart, referred to as cardiomyopathy, or both in combination. ATTR amyloidosis can also be acquired, known as wild-type ATTR amyloidosis. This causes a predominant cardiomyopathy and is an increasingly recognized cause of heart failure. The exact prevalence remains unknown. Both conditions cause debilitating symptoms with a high burden of disease for patients and caregivers. Intellia's Phase 1 study is in the hereditary ATTR amyloidosis with polyneuropathy population. This is the population in whom knockdown of TTR protein has been shown to have clinical benefit. It is also the population that's most likely to provide clear data on safety whilst establishing a dose effect for NTLA2001. However, there's no conceivable reason to believe that the effects of NTLA2001 treatment will not translate to the entire ATTR amyloidosis population. Let me explain why. In every type of amyloid that's ever been treated, there has been a very strong correlation between the degree of knockdown of the relevant protein and clinical outcomes. The explanation is very simple. The turnover of amyloid deposits depends on the equilibrium between the rate of amyloid production and the rate of amyloid removal. The rate of removal is, by definition, very slow, such that in an untreated individual, the rate of amyloid production exceeds the rate of removal, amyloid accumulates, and results in clinical progression and death. The more one knocks down the concentration of the relevant protein, in other words, the rate of amyloid production, the greater the equilibrium in favor of amyloid removal. Initially, when we started treating light chain, also known as AL amyloidosis, we thought that a greater than 50% reduction in the amyloid-forming protein, in other words, the light chain, was sufficient because outcomes were better than in patients with no knockdown. Many years later, however, we now know that what hematologists need to aim for is what they call a complete response. In other words, 100% reduction of the amyloid-forming protein. If you have a look at this graph, at the bottom, the line at the bottom is patients who've achieved a less than 50% reduction in the amyloid-forming protein. The next graph up is patients who've achieved a greater than 50% reduction but less than 90% reduction. The next line up is patients who've achieved what hematologists call a VGPR, which is a greater than 90% reduction in their light chain. And the top line, the best survival, is amongst patients who achieve what hematologists call a CR, or a complete response. Exactly the same is true in AA amyloidosis, a different form of amyloidosis. This table shows the relative risk of death in patients as the octiles of serum amyloid A protein, that's the protein that is the amyloid-forming protein in this type of amyloid, decreases. And you can see that the relative risk of death decreases progressively as the SAA concentration decreases. In ATTR amyloidosis, we see exactly the same trend emerging, although the data at this point are less mature, since TTR knockdown has only been possible in the last few years. This, however, shows the relationship between degree of TTR knockdown and change in neuropathy score. We see that lower serum TTR has been observed to be associated with better subsequent control of symptoms from disease. So taken together, these data form the basis for the hypothesis that knockout of the TTR gene to achieve deep TTR reduction may halt and potentially reverse this disease, and the conviction behind my involvement with this study. I will now show the presentation from Saturday's 2021 PNS annual meeting. It's a privilege to share with you interim data from the first in-human trial of an in vivo CRISPR-Cas9-based gene editing therapy, NTLA2001, designed to edit the TTR gene in patients with transthyretin amyloidosis. These are my disclosures. As I said earlier, the main therapeutic strategy successful in all types of amyloidosis that are amenable to treatment is to reduce the concentration of the circulating amyloid-forming protein, in this case transthyretin. The gene silencing therapy patisiran reduces serum TTR concentration by approximately 80% and has been shown to benefit neuropathy in hereditary ATTR amyloidosis. However, gene silencing therapy requires chronic administration, and one would expect greater TTR knockdown to achieve better clinical outcomes. There is therefore an unmet need for better treatments in ATTR amyloidosis. Editing of the TTR gene, which has the potential to provide permanent and profound TTR knockdown after a single infusion, is a particularly attractive treatment strategy. NTLA2001 is a CRISPR-Cas9-based in vivo gene editing therapy. It consists of a single guide RNA molecule that targets the human TTR gene and a human-optimized mRNA sequence of strep pyogenes Cas9 protein encapsulated in a lipid nanoparticle, or LNP. Following intravenous administration of the LNP, it's transported directly to the liver, where it's taken up via the LDL receptor on hepatocytes. After endocytosis and release into the cytoplasm, the Cas9 mRNA is translated, producing the Cas9 enzyme, which interacts with the single guide RNA to form the CRISPR-Cas9 ribonucleoprotein, or RNP complex. The RNP complex enters the nucleus, and a target-specific 20-nucleotide sequence of the 5' end of the guide RNA binds to the DNA double helix at the target site. This leads to precise cleavage in the targeted TTR gene sequence. Endogenous DNA repair results in introduction of insertions or deletions of bases in the target site. These frame-shift alterations reduce functional target gene mRNA and consequently production of the target protein. The plot here shows the in vitro therapeutic index with effects of concentration of the guide RNA on the percentage TTR gene editing by NTLA2001. This was performed in cultured primary human hepatocytes. The vertical line here marks the concentration at which we routinely observe 90% reduction in TTR protein production, the EC90. Above the EC90, NTLA demonstrated saturating levels of TTR gene editing accompanied by greater than 95% reduction in TTR protein. An important question the scientific team asked before bringing this agent to clinic was, what is the level of off-target editing at these concentrations? The team performed extensive genome-wide screening with computational and laboratory-based techniques to detect and validate the likeliest sites for potential off-target editing. These methods identified 7 sites, all in non-coding regions. The experiment shown here compares the rate of on- and off-target editing following treatment with NTLA2001. What you see marked down here by the colored lines, each representing one of the 7 sites, is that there was no detectable evidence of off-target editing, even at concentrations of NTLA2001 threefold greater than the EC90. Preclinical in vivo studies were conducted in cynomolgus monkeys using a surrogate LNP incorporating a single guide RNA specific to the monkey TTR gene. This graph shows the mean percentage reduction from baseline in serum TTR protein concentration in monkeys receiving the LNP at doses of 0, 1.5, 3, and 6 milligrams per kilogram of the RNA cargo on day 0 and followed for 367 days. Vertical lines represent standard deviations across the 3 main animals in each group. Note a single dose at 3 milligrams per kilogram achieved a durable reduction in circulating TTR protein of approximately 95% after day 28. Safety studies established a dose range at which no adverse effects of NTLA2001 were observed, with near complete sustained reduction in serum TTR following a single administration. At higher doses, the adverse effects in the monkeys were dose-dependent, transient, reversible, and monitorable. The first-in-human study with NTLA2001 is a 2-part, open-label, multi-center study in patients with hereditary ATTR amyloidosis and polyneuropathy. Part 1 is a single ascending dose design with a minimum of 3 patients in each of up to 4 dose escalation cohorts. The starting dose in humans was 1/10 of the allometrically scaled dose at which no adverse effects were observed in the monkey. The recommended dose selected in Part 1 will be administered to a dose expansion cohort in Part 2. The primary objectives of the first human study are to evaluate safety, tolerability, PK, and PD, the latter by serial measurements of TTR levels. I will report data on the first 6 patients from the first 2 dose escalation cohorts included in Part 1. Bear in mind, participants received a one-time infusion of NTLA2001 on day 1 of the study. Here are the demographics of these first 6 patients, 3 in each dose cohort. Age range from 46 to 64. 3 different pathogenic TTR mutations were represented, and 3 patients received prior diflunisal therapy. All had early-stage polyneuropathy, and despite presence of cardiac ATTR amyloid by imaging in 5 of 6 patients, accompanied in some by elevated NT-proBNP, they were all NYHA class 1. Median time from diagnosis to treatment was 2.5 years. NTLA2001 was generally well tolerated in the acute phase, with AEs all being grade 1 and mostly unrelated to the treatment. 1 patient had a mild infusion-related reaction, but this did not require dose interruption. There were no SAEs. There was no evidence of liver toxicity or coagulopathy, and laboratory excursions were clinically insignificant. Serum TTR reduction was observed by day 7 and deepened further by day 28 in all patients at the first dose level of 0.1 milligram per kilogram. The reductions ranged from 47 to 56%. And the reductions were dose-dependent, with greater reductions detected in the 3 patients who received the higher 0.3 milligram per kilogram infusions. Here, the reductions range from 80 to 96%. At day 28, mean TTR reduction from baseline in the 0.1 milligram per kilogram cohort was 52%, and in the 0.3 milligram per kilogram cohort was 87%. In conclusion, systemic administration of NTLA2001 in patients with hereditary ATTR amyloidosis and polyneuropathy caused a profound and dose-dependent reduction in serum TTR protein concentration. Of note, maximum reductions were 56% and 96% respectively in the 2 dose cohorts. NTLA2001 treatment was well tolerated in the acute phase, with all AEs being of mild severity. Further dose escalation is ongoing in this first-in-human study with the aim of achieving greater reduction in TTR than that provided by currently available agents, which is expected to translate into improved clinical benefit for patients. For example, improvement in autonomic symptoms, better mobility, and fewer cardiac hospitalizations for heart failure. This is the first demonstration of CRISPR-based in vivo gene editing in humans. These findings provide proof of concept for a promising new therapeutic strategy. I would like to acknowledge the patients, the contract research organizations involved in the trial, my co-investigators at the National Amyloidosis Center, Richmond Pharmacology and the University of Auckland, and of course the many members of Intellia Therapeutics and Regeneron Pharmaceuticals who were responsible for the development of NTLA2001. Now I'd like to turn the call over to David LeBeau.
Thank you, Dr. Gilmore. Your leadership in this trial has been critical for its success. We also appreciate your vision for 2001's potential to improve on patients' health outcomes compared to current standards of care by achieving deeper TTR reductions. Before outlining next steps for this program, I'd like to spend a minute reviewing our objectives for 2001 and specifically what we had hoped to achieve with this readout. Since initiating the study, we suggested that this readout would share data that is consistent, meaningful, and interpretable, although interim. And with this report occurring during the dose escalation phase of the trial, we're looking to demonstrate the initial safety and activity profile of the drug. We hope to see a consistent dose response and good tolerance of the LNP formulation as indicators of proof of concept for our platform and important steps towards identifying the recommended dose for 2001. As you can see, we have achieved our goal. We're highly encouraged by these interim results, which to date show that 2001 was associated with only grade 1 adverse events in the first 2 dose cohorts. Equally important, we have already achieved a deeper TTR protein reduction on average in the second cohort than the current standard of care. We anticipate that a higher dose may further drive TTR levels consistently lower than any other therapy has been able to achieve, with expected corresponding clinical benefit. Based on the safety and tolerability data observed to date, we are continuing to dose escalate. Taking these early results from part 1 of our study, together with our robust demonstrations in the preclinical experiments, we believe 2001 has the potential to be a first-in-class, single-dose treatment for patients with ATTR amyloidosis. In terms of next steps for this program, we're pleased to share that we are actively enrolling Cohort 3, where each subject will receive a single dose of 2001 at 1 milligram per kilogram dose level. We plan to provide additional data from Part 1 of the study at a scientific or medical conference in the second half of the year. In this next update, we plan to share data further along in our dose escalation, as well as additional durability data beyond 28 days from the initial cohort. Once we identify the recommended dose, we plan to initiate Part 2 of the trial, a single-dose expansion cohort. In this portion of the study, 8 subjects will be administered the recommended dose identified in Part 1, with the objective to further characterize the activity of 2001, including an initial assessment on clinical measures of neuropathy and neurologic function, as well as additional safety data. We expect to advance into Part 2 of this study later this year. Our goal then is to expand the study and to engage with regulators, including the FDA, to move into later studies in which we aim to include both polyneuropathy and cardiomyopathy patients. This is an exciting first look as we continue to make steady progress in the clinical study, and we look forward to keeping you updated on our progress. Before passing this call along, although the trial is ongoing, we'd like to express our gratitude first and foremost to all the patients and their caregivers for their participation in the study, and also to our investigators and their site support staff. With that, I'd like to turn the call over to our Chief Scientific Officer, Laura Sepp-Lorenzino, who will review today's update in context of our platform and broader in vivo pipeline.
Thank you, David. These results are significant in terms of what they mean for ATTR amyloidosis patients. We're equally excited about what they may imply for our efforts beyond NTLA2001. As John noted, Intellia's platform was built intentionally to enable modular solutions that would support the broadest possible therapeutic application for our editing and delivery technology. In the in vivo setting, we pursued systemic delivery to achieve this ultimate objective. In particular, we selected a lipid nanoparticle-based solution for the numerous advantages it presents, including transient expression, large cargo capacity, and the potential to redose, and also because it introduced a rapid and reproducible path to clinical development once we demonstrated proof of concept. This interim update offers just that. We believe we have unlocked the lever. Because many genetic diseases involve genes that are active in the liver, enabling systemic CRISPR delivery to this organ should allow us to develop treatments for multiple diseases, both rare and prevalent. These results demonstrate we can deliver it to hepatocytes, and by swapping out the target site portion of the guide RNA, we're able to precisely edit different genes. We can therefore accelerate the development of additional in vivo programs with increased probability of technical success. This includes several indications we've already explored preclinically and numerous others, and especially applies to follow-on in vivo knockout programs such as NTLA2002, our candidate in development for hereditary angioedema, or HAE. NTLA2002 leverages the same LNP and RNA cargo, now targeting the KLKB1 gene in the liver, to permanently reduce plasma kallikrein protein and activity, a key mediator of the disease. This approach is designed to provide continuous suppression of kallikrein activity and eliminate HAE attacks. Today, alongside the NTLA2001 interim results, we're very excited to share that we have filed our first CTA for NTLA2002 in New Zealand. And on the basis of learnings from NTLA2001, including the results discussed today, we expect to move faster and begin evaluation at the higher dose with NTLA2002. We plan to enroll our first patient in a phase 1 study of NTLA2002 by year end. Further, we believe this interim data has applicability to our targeted insertion and consecutive editing strategies since it utilizes the same CRISPR delivery system. We continue to advance this work internally and in partnership with Regeneron, and look forward to evaluating these hypotheses in the clinic as well. Finally, as this slide outlines, the next wave of growth for our in vivo efforts is to extend the applicability of our delivery platform and editing tools to pursue disease targets across multiple tissues. Once again, the data presented today offers meaningful validation for our modular platform and programs well beyond NTLA2001. It paves a path forward, de-risking our LNP delivery platform, accelerating our in vivo pipeline, and offering a springboard to set up our long-term vision for Intellia. And with that, I'll turn the call back to John for closing remarks.
Thank you, Laura and David, and especially to Dr. Gilmore. We appreciate the time you spent with us today. As you can see, observations so far from our phase 1 study of NTLA2001 are very encouraging, offering support at both program and platform levels. For patients with ATTR amyloidosis, we believe these interim results point to NTLA2001's potential to halt and reverse their disease with a single dose. We believe this demonstration of systemic in vivo genome editing substantially de-risks our LNP platform and accelerates our in vivo pipeline. And we're proud to lead the way into this new era in medicine in which curing genetic disease may be within reach. And we're just getting started. We're executing against the ambitious goals and strategic priorities set out at the beginning of this year, having now checked boxes for clinical validation, advancement of our full spectrum pipeline, and continued platform innovation. And as our investment in R&D matures into a broad pipeline of clinical candidates, we remain steadfast in our commitment to harnessing CRISPR's full therapeutic potential. Turning our attention to the second half of this year and beyond, we hope to carry this momentum forward as we close in on a number of key upcoming milestones. For NTLA2001, we plan to provide additional data from part 1 of this study at a scientific or medical conference this year. And once the recommended dose has been identified, we expect to initiate part 2, our single dose expansion cohort, later in 2021. For NTLA2002 or HAE, now that we've filed our first CTA, we plan to enroll our first patient in a phase 1 study by the end of this year. For NTLA5001 for AML, we are on track to submit an IND or equivalent regulatory filing in mid-2021. And across our R&D efforts, we remain on track to nominate at least one new development candidate this year, along with the nomination of our first allogeneic development candidate by the first half of next year. As Laura stated, today's update provides a springboard from which to advance our long-term vision for Intellia. As we look ahead for our in vivo approach, our interim data supporting the successful delivery to and inactivation of a gene in the liver opens the door to targeted gene insertion and ultimately the next wave of therapies targeting diverse tissues across multiple diseases with our proprietary delivery technology. Further, we continue to advance CRISPR cell engineering in the ex vivo setting. This includes introducing a differentiated allogeneic solution, as well as applying our proprietary base editing technology introduced earlier this year as we continue to expand our pipeline. Taken together with today's announcement accelerating our in vivo strategy, we have a path forward to advance 1 to 2 new programs per year. Without a doubt, this is an exciting new chapter for Intellia, for genome editing, and most importantly for patients. We look forward to sharing more from NTLA2001 and other parts of our pipeline as we continue to work toward fully realizing the promise at the heart of the genomic revolution. Operator?